Welcome to Biology Made Easy. Today we are discussing cell surface membranes. You have a cell here, it has a membrane here, and there are organelles in the cell. They also have membrane. The slight difference between the membranes of the organelles and the cell surface membrane we'll mention along. But we are discussing the fluid mosaic model of self-membrane by Singer and Nicholson, 1977. Now, mosaic means that the cell membrane has a mixed structure, and we will look at the components of the cell membrane. Now, the components primarily is phospholipid, and so we'll look at the structure of the phospholipid. The phospholipid has a phosphate head and fatty acid chains. So this is the phosphate and this is chains of lipid. Now the phosphate is hydrophilic, meaning water loving or attracting water, and the fatty acid chains are hydrophobic, means water repelling. And the phospholipid is arranged in bilayer. Now, the lipids has kinks or bends in their structure because chains of fatty acids. Some have double bonds in them. When there are double bonds, then there are kinks, bends, like this. Unsaturated lipid. This is saturated lipid. The lipid chains are straight, but these ones have double bonds in it, so they have kinks or bends. And so because they have kinks or bends, the lipid chains are not packed closely together. And so the bilayer is kind of fluid because the fatty acid chains has bends. And not only that, the phospholipid, some can turn or rotate on their long axis, and some can also flip the tails of lipids. And so the phospholipid layer is fluid. There's sideways movements in the membrane, and it's been found that there are compartments of movement. The function of the phosphate bilayer is it binds the cytoplasm into a unit which is called cell, and then it is selectively permeable. We'll talk about it in our next lesson, the adaptations, and so explain why the membrane is selectively permeable. Then you see we said the membrane is fluid, and so it's soft and flexible so that it can not break when it expands. Now, embedded in the bilayer of phospholipid are cholesterol and membrane protein. Now, the cholesterol is steroid, and in animal membranes we have cholesterol, but in plants we have phytosterol, which is a kind of a sterol in plants. But in animal membranes, we have cholesterol. And the cholesterol has their polar hydroxyl ends very close to the polar phosphate. You see, the phosphate is hydrophilic, and the polar hydroxyl end of cholesterol is lined up at the polar ends. So this is cholesterol. We can also have some cholesterol in here. So we have cholesterol in the membrane. And the function of the cholesterol is the cholesterol maintains the permeability of the membrane and then keeps the temperature of the membrane constant. It prevents the membrane from freezing, because when the temperature is high and the membrane has a tendency of expanding so that the pores are big, the cholesterol resists that. And then when the temperature is low and freezing, the cholesterol prevents the membrane from freezing. So the cholesterol maintains the permeability of the membrane and also keeps the temperature constant, prevents the cell membrane from freezing, which is very important to organisms that live in very cold areas. We have membrane proteins. It means proteins in the membrane. The membrane protein can be one protein or a collection of proteins, and the proteins are put together in such a way that there will be a pore or a channel in the protein. So if I have a protein like this, and a protein like this, and the protein has a channel. This is a channel or a pore. So there are different types, but that is membrane proteins. They are arranged, and the pore or the channel is hydrophilic. There are different types of proteins. One type is integral, and the integral protein is a protein that spans the whole length of the membrane. So I have an integral protein, it spans the whole length of the membrane. There are two types of integral proteins. One is called channel protein, one type, and another type is carrier protein. This is a channel protein. This is a carrier protein. The channel protein, their structure is fixed structure with a channel or a pore, hydrophilic pore or channel, and substances are transported through the pore or channel. The shape is fixed. But the carrier protein, the protein is such that as it transports substances across, it changes shape. So at one time, the shape will be like this, and it's bringing a substance across, and it will open for the substance to drop. So the carrier proteins changes shape. This is transporting a substance, is the red bit. It uses energy, ATP, to do that. And as the substance is brought in here, it changes shape, releases the substance, and then the shape goes back. And the changing of shape or conformation is very rapid, more than 100 cycles per second. They change here. Very rapid, transporting substances. That is integral proteins. The integral proteins, they span the length of the membrane, they are fibrous, and they are permanently embedded in the membrane. Then there's another type of membrane protein called peripheral protein. The peripheral proteins do not span the full length of the membrane. The peripheral proteins are found on the outer surface of the membrane. This is a peripheral protein, and this is a peripheral protein. They are found on the outer surface of the membrane or inner surface of the membrane. And the peripheral proteins, they are not permanently associated with the membrane. They are affected by the pH or the anionic concentrations, and also they can easily be removed by certain solvents. There's also protein called alpha helix proteins. Alpha helix protein, they are proteins whose amino acids are arranged in helices. So I have protein like this, it's an alpha helix. And these alpha helix proteins, they can be integral, that is, they span along the length of the membrane, or they can be peripheral. So we can have an alpha helix proteins as a peripheral membrane. So we have alpha helix protein, and then we have globular protein. The globular protein, they are kind of spherical, they are water soluble, and alpha helix protein and globular protein, they can be integral or peripheral. So these can be integral or peripheral. It means if I have a globular protein, also somewhere in there, there's a globular protein, I can have the globular protein that is associated with the outer surface or inner surface, or a globular protein that spans the length of the membrane, that is integral. So they can be integral or peripheral. That is globular protein and alpha helix protein. Now, globular proteins are spherical, compact, and they are water soluble. They're not like the integral proteins that are fibrous. And then we have other components which I label as 4. We have the glycoproteins and glycolipids. So the glycoproteins, we have glycoproteins and glycolipids. The glycoproteins are chains of carbohydrates. This is a chain of carbohydrates. You have chains of carbohydrates attached on protein. That's a glycoprotein. And then the glycolipids are also chains of carbohydrates attached on lipid, the lipid part of the membrane. And these glycoprotein and glycolipids, they are only found on the outer surface of the membrane where there are carbohydrates area looks like antennae, and they are found sticking onto the extracellular surface. This is the surface extracellular, this surface in the cytoplasm. So these carbohydrates, these glycoproteins and glycolipids, they serve as markers. They have fillers, especially the glycoproteins that produce antibodies against bacteria and foreign matter. And both glycolipids recognizing similar cells or cells that are different. So glycoproteins and glycolipids, these form some kind of antennae on the surface of the membrane, watching so that they can protect the cell from non-cell things, and also they help similar cells to stick together to form tissues. That's the function of glycoproteins and glycolipids. Notice cells and bodies that are foreign work with the immune system, right, to protect the cell. The peripheral proteins, you have these also as integral peripheral proteins. They link up with cytoskeleton in the cytoplasm. So these are cytoskeleton, this cytoskeleton, cytoskeleton, these are fibrous structures. These peripheral proteins, they link up with cytoskeleton and make the cell firm, so they give structural support. Our neat cell membrane here. Let's quickly look at the functions of the integral proteins and the peripheral proteins. Integral proteins here, channel protein, you have carrier proteins, these two are carrier proteins, see how they go. This is alpha helix protein. The integral proteins, their function is they are transporters. They transport nutrients, waste, various chemicals, ions that are needed for processes inside. They transport it across the membrane. They are also receptors where hormones bind on the surfaces. They are receptors. And if hormones bind on the surfaces, the hormones will trigger a chain of reactions inside the cell. They are also enzymes. You know what enzymes do? They speed up processes, so some of the proteins serve as enzymes. And then signaling also means that they are affected by proteins and other physical structures like light that also see to perform a chain of reactions inside the cell. That's signaling. Now, peripheral proteins do almost the same things except transporting substances. But the peripheral proteins, you can have globular proteins that do a little bit of transport, and they also will do a little bit of transport. But then these are their functions: receptors, enzymes, signaling, and then structural support, because the peripheral proteins link up with cytoskeleton to firm up the cell and the membrane. Now, you know, this is the cell surface membrane because on its outer surface, you have the glycoproteins and the glycolipids. As we said, the membrane structure, this is the cell surface membrane. The organelles also have membranes. Organelles like mitochondrion, they also have membrane. Even endoplasmic membranes, vacuoles, they all have membranes. The membranes are just like this, with the exception that they will not have glycoproteins and glycolipids that stick into the intracellular fluid, guarding, so to speak, the cell. This is what we have in this lesson. So till we meet in our next lesson, if the lesson has been fruitful, please give us your likes. Do kindly subscribe to our channel if you haven't subscribed already. Thank you very much. Goodbye.